Hello, my name is Barbara Delgado. Welcome to my presentation on this week's module reading, Increased Tumor Homing and Tissue Penetration of the Filamentous Plant Viral Nanoparticle Potato Virus X, a journal article from a familiar friend, Case Western Reserve University. In this presentation, I will be discussing the terminology, traditional viral nanoparticles used in tumor homing, the problem with them, and the proposed solution from this research. This research was inspired by the recent data that indicated advantages of filamentous nanomaterials for in vivo medical applications. Filamentous is simply the description of the shape of the nanomaterial. It is elongated thin series of cells attached to one another, or a very long thin cylindrical single cell, like a filament. Also, if you called it a worm-like nanoparticle, it would sound creepy. Nanoparticles have shown the ability to carry large drug payloads and also have the ability to be programmed to deliver to specific target sites. This makes them vital vessels for drug administration for cancer and cardiovascular disease. The shape and elasticity can significantly impact their ability to do their job in vivo. Filamentous nanomaterials have been shown to have superior ability to assist in drug absorption and distribution and tumor homing properties. But the majority of research that is currently being performed are using spherical and elongated low aspect ratio materials. The aspect ratio is referring to the geometry of the material. The higher aspect ratio, the more filament-like the object is. In the image below, shows some typical viral nanoparticles used in research. The majority of them are spherical in shape, except the tobacco mosaic virus, which was mentioned in the article, where it has a cylindrical geometry. So what's the problem? Why can't we synthetically create high aspect ratio nanoparticles? It is difficult to create these high aspect ratio geometry on a nanoscale level using synthetic materials. The technology is just not there. This is mainly due to the current technology's ability to control chemical and biological reactions at that scale. So what is the solution? These researchers have found a way to engineer viral nanoparticles, or VNPs, from plants for imaging and drug delivery. They decided that since engineered filamentous nanoparticles are nearly impossible to make synthetically, why not take advantage of the best engineer in the universe, nature? Although certain viruses can kill you, they are good at their job, which is carrying and delivering stuff quickly and effectively through an organism. They also come in many shapes and sizes, which is what the researchers are interested in, including on the nanoscale level. It has been proven that viral nanoparticles are safe from a human health perspective, since they are not pathogenic to humans or animals. These researchers focus on high aspect ratio filamentous plant viral nanoparticles. The two viruses of interest are the Cowpea mosaic virus, which has been traditionally and extensively studied and developed in vivo, so its properties are well understood, and also have a spherical shape, as shown in the bottom image. The two viruses, and the second virus is Potato-Virus X. Potato-Virus X is an emerging filamentous viral nanoparticle platform being developed for nanomedical applications. These two viruses had their biodistribution and tumor homing properties compared. The two viruses were tested in preclinical animal tumor models in this article. And as you can see here on the top image, the potato virus X is extremely, I call worm-like, but filament-like, versus the cowpea, which is more spherical. After extracting the viruses from an injected plant, the virus concentration in the plant extracts what was determined. The virus concentration in the plant extracts was determined by UV visible spectroscopy. An example shown on the top right. The structural integrity of the nanoparticles was further confirmed using transmission electron microscopy and size exclusion chromatography. An example shown on the bottom right. A near-infrared dye was used to confirm the concentration ratio of dye moieties per viral nanoparticles based on the UV visible spectrum using fluorescence. They used Beer-Lambert's law, a familiar friend to us. Tumor homing and tissue penetration properties of the two viruses were evaluated with two tumor models. Using confocal microscopy, the two tumors that were injected in each virus were collected, sectioned, and imaged. A confocal microscope is shown on the bottom left. An example of chromatography is shown on the bottom right. So what are the results of this? Microscopy showed that both the potato virus X and cowpea mosaic virus accumulated in solid tumors after administration. The localization was different between the two viruses. The potato accumulated in the center of the tumor. This was seen in both types of tumors using the potato virus. The cowpea mosaic virus did not penetrate as deeply as the potato virus X. Cowpea mosaic virus localized through the tissue with no apparent increase in the tumor center. Looking at the tumors under a microscope, the potato virus X spread consistently throughout the tumor tissue and penetrated into areas in which the cowpea mosaic virus was not apparent. This adds to the growing proof that indicates that filamentous structures have better diffusion rates in fibrous matrices like tumors. So what's next? There is a great potential in this new method of biologically inspired drug distribution. In the article, they noted that they were aware that charges of particles also have an effect on the ability of the nanoparticles' ability to penetrate and deliver. Also, it seems that potato virus X is a strong candidate for further research in cancer delivery nanoparticles due to its geometry. That is the end of my presentation. I look forward to reading your comments and questions. Have a great week.